Hello everyone, and welcome to the next problem solving session for the course Novel Technologies for Food Processing and Shelf Life Extension. So, in this week, we'll be solving problems related to week 8. We'll begin with the problems. So, if you guys have any doubts in the session, you can either mail me or post it in the discussion forum, and we'll take it up in the next class and discuss that as well. So, question 1. Which of the following is the strongest fiber? A. Nanotube, B. Nanocell, C. Nanofibers, and D. Nanocomposites. So I'll be discussing the answers along with this as well, since people are not here for the live session. So the strongest fiber among these four is the nanotubes. So nanotubes, as you can see in this picture, it is made up of hexagonal shaped structures, and they are actually like sheets with a honeycomb or hexagonal shaped structures, and they are rolled into a tube. So this rolling of the tube makes it very stable, strong, as well as a flexible structure. So as you can see here, it's very tiny fiber. It's only of the size of somewhere around 1 to 10s of nanometers. And these nanotubes are found to be 10 to 100 times stronger than that of the steel. So nanotubes are basically sheets that are rolled into a tube-like structure. Nanotubes are basically sheets and they are rolled into tube-like structures. And these tubes makes it one of the most strongest fibers present. So nanofibers and nanocomposites are used for different other purposes. So nanofibers are also fiber-like structures, but they are not as strong as a nanotube. And nanocell is often used in, it's more like a sensor kind of this thing. Nanocellulose are often used in, like, say, TVs or other kind of electrical circuits as well. And nanocomposites are nanomaterials where you have more than one phase. So you have more than one phase that come together and impart a much more better properties than that of a single phase. So multiple phases come together to impart much better properties in case of the nanocomposites. And in the nanocomposite, at least one of the phases is in the nanometer range. The answer is option A. Moving to question 2. Choose the correct option which depicts the right chemical formula for the respective tocopherols presented in the undermentioned figures and tables. So before we go forward, let me explain what a tocopherol is. So often oils undergo oxidative rancidity and other processes that result in changes in its flavor, changes in its color, etc. So this happens because of the oxidation processes of the especially the double bonds present in the oil. So because these double bonds undergo oxidation processes, it is necessary to add certain antioxidants in the oil to prevent this oxidation process. So one of the antioxidants that you add are tocopherols, and tocopherols are majorly are of four types. So this is the basic structure of the tocopherol, where you have R1, R2 and R3. So these are different functional groups that can be present in the tocopherols. So based on what is present on R1, R2 and R3, the name of the tocopherol would change. So in this question, what should be R1, R2 and R3 for alpha tocopherol, for beta as well as gamma tocopherols? So you have the options here. So as you can see, it is majorly hydrogen or the methyl group that is kind of interchanged across R1, R2 and R3 to give it three different kinds of tocopherols. So in this case, for alpha tocopherol, the R1 should also be a methyl group. So in alpha tocopherol, both R1, all the three, R1, R2 and R3 are the methyl groups. And in case of beta tocopherol, you have the second group as your hydrogen. So it will be CH3 here, and it will be H here, and it will be CH3 here in case of beta tocopherols. And in case of gamma tocopherols, you have a hydrogen here at R1, you have R2 as CH3 and R3 also as CH3. So that is for alpha tocopherol, that is the first one, you should have a CH3, and in case of beta tocopherol you should have a hydrogen, In case of gamma tocopherol, you should have a hydrogen here and a methyl group here. So basically the answer is option C. So alpha tocopherols are also known as 578 trimethyl tocol because it has all the three methyl groups in place of R1, R2 and R3. Whereas in case of beta tocopherol, only R1 and R3 are methyl groups. That is, only the 5th and 8th position have methyl groups. So it is known as the 5,8 dimethyl tocol. And in case of gamma tocopherol, you have R1 as H, only R2 and R3 as the methyl groups. So you have 7 and 8th position having the methyl groups. So R2 and R3, so because of R2 and R3, gamma tocopherol is also known as 7,8 dimethyl tocol respectively. So the answer to this question is option C. So question third. Make proper combinations of the antioxidants and the category to which they belong. So first in column 1 you have gallets, second is BHT, third is EDTA and fourth is ascorbate. And you have the category of the antioxidants on in the second column. So the first are hindered phenols, second is oxygen scavengers, third are phenols and fourth are metal chelators. So hindered phenols and phenols form the primary antioxidants, whereas the rest, the oxygen scavengers and the metal chelators, form the secondary antioxidants. So if you look into each of these compounds, so the food antioxidants can be classified into primary and secondary antioxidants. In case of the primary antioxidants, both phenols and hindered phenols form the primary antioxidants. So galates come under phenols and your BHT comes under hindered phenols. So the difference between phenols and hindered phenols is that hindered phenols will have a bulky functional group apart from the phenolic group. So phenols will just be your phenolic groups, and hindered phenols will have a much more bulkier functional group apart from the phenols. So galates come under phenols, BHT comes under hindered phenols. So both of these are primary antioxidants. And in case of your secondary antioxidants, EDTA, as we all know, can chelate metal ions like magnesium and all. So they can often chelate divalent ions and metals. So EDTA comes under metal chelators, whereas ascorbyl comes as oxygen scavengers. So all these, when you add all these antioxidants inside your lipids, it prevents these oxidation processes. It prevents these oxidation processes by different methods. One of them is the metal chelation, the other like oxygen scavenging and so on. The answer is galates to phenols, BHT to hindered phenols. So 1 stands for 3, 2 stands for 1, EDTA stands for metal chelators, and ascorbyl stands for oxygen scavengers. So the answer is option D. So question 4. Which of the following microencapsulation related statements are true? So 1. Hygroscopicity of material can be decreased by microencapsulation. Second, objectionable taste and aroma can be masked by microencapsulation. Third, encapsulation of sodium chloride are done using spray drying. And fourth, molecular inclusion of the flavor volatiles in beta cyclodextrin molecules is the least effective method for protecting the aromas. So one of the, this is basically the roles of microencapsulation that is being asked in the question. So basically hygroscopicity of a material means if you leave a material outside in the air, it often absorbs water that is present in the surrounding atmosphere, because of which the material will now become wet and soggy, and this is not preferred in a lot of food products. So in order to prevent the material from being exposed into air, often what is done is the material is microencapsulated. So if you encapsulate the material with say some polymer or so, its exposure to air will decrease, and hence your hygroscopicity of the material can also decrease. So this is followed for food products like sodium chloride, where if you leave salt outside for a long time, it will start to absorb water. So the hygroscopicity of the material such as sodium chloride can be reduced by microencapsulation. So yeah, microencapsulation can also be used when you have food ingredients that have objectionable taste and aroma. So what often happens is you have some bitter substances or so, or if the ingredients that you add kind of interact with the other food components and then gives an objectionable taste or aroma, then the best thing to do is to microencapsulate them. That is to separate them from other ingredients by having a coat around those substances. So if you have a coat around those substances, that objectionable taste and aroma is not that obvious because of microencapsulation. So the objectionable tastes and aromas of popular nutritional ingredients such as soy extracts, bitter herbs, omega-3 oils can be masked by microencapsulation. So third, the encapsulation of sodium chloride is not done using spray drying. It is done by a method known as fluidized bed coating and extrusion. So these they are two different methods that are used for encapsulation of sodium chloride. So we will discuss both the fluidized bed coating and extrusion in the later slides. So for sodium chloride, these two are the most common methods for encapsulation. And fourth, the molecular inclusion of the flavor volatiles in beta cyclodextrin molecules is actually the most effective method for protecting the aromas. So if you have volatile substances that give it the aroma for the beta cyclodextrin, and in order to protect that aroma, you can often do this microencapsulation that will keep the aroma safe within the molecules. So amongst all my existing microencapsulation methods, molecular inclusion of the flavor volatiles in the beta cyclodextrin is actually one of the most effective methods for protecting the aromas. So 1 and 2 are true, whereas third and fourth are false. So the answer is A. So both the hygroscopicity of the material can be decreased and exposure to objectionable taste and aroma can also be decreased with the help of microencapsulation. So moving to question 5. This is actually the extrusion method that I had talked in the previous slide. So in the undergiven figure, choose the correct sequence of the material to be added in the extruder for encapsulation. So you have 4 different components involved here. One is water, second is wall material, third is additives, and fourth is core material. So the core material is your desired component that you want to encapsulate. That will be inside the coat. And a wall material is what will polymerize around the core material. So you have an inside inner core and you have an outside wall material. So both of these you need to add along with certain additives to help it cross polymerize and then set. So it has to harden around the core material also. So in order to harden and to make the wall material stable, you often add additives. Uh, and these are the three major things. These are the 4 major raw materials required for an extrusion process that involves microencapsulation. So the first thing that you add in an extrusion, so you have multiple holes here, and there's a specific method or a specific sequence in which you add these raw materials. So the first thing that you add is the wall material and the required amount of water at the feed end. So 1 and 2 stands for the wall material and the water. So both of them will first mix and then you add the core material. That is the material that you want to encapsulate. So third is where you will add your core material. And because of this extrusion process, both the wall material, the water and the core material will start mixing. And then you also add additives to stabilize the wall material. So after this, once you add all the 4, that is first is wall, second is water, then third you add the core material, and fourth you add the additives. So once you add all these raw materials, what happens is your emulsion will start moving from this direction to this direction, and it will pass through a die that will give it a certain structure. So this micro capsules are formed once the material passes from the die into the outside environment. The extruded that comes out of the die and then cut to bring it down to very small sizes. So once the extruded comes out of the die, you use a cutter to cut it, and that brings about the sizes of a microcapsule. So the answer is option C. First is wall material, second is water, third is a core material, and fourth are additives. Okay. Moving to question 6. Which of the following is most commonly used for micro encapsulation in a food industry? So you have 4 different techniques, all 4 are used, but one of them is the most commonly used because of its high efficiency and flexibility and so on. So first is a spray dryer, second is a freeze dryer, third is vibrational nozzle, and fourth is the pan coating. So multiple methods are used for the process of micro encapsulation. First is a spray dryer, which is actually the most commonly used method for encapsulation in the food industry, and this is because it is very economical and it is very flexible. So based on the kind of particles that you want, uh, you can actually control a lot of parameters to get a desired micro encapsulation efficiency and so on. So in case of spray dryer, what you have is you have an atomizer or so that will spray all your particles in the presence of both the core and the wall material. And when your wall material and your core materials are sprayed into the dryer, because of its small size, what will happen is the wall material will now surround the core, that will harden and then give you the micro encapsulated product. So what happens in spray drying is that you prepare an emulsion, then you homogenize the dispersion, and then you atomize it into the chamber. And because of this atomization, now you'll get very small sizes of micro encapsulated product. Next is the freeze drying process, so spray drying is actually the most commonly used for the micro encapsulation. So that is your answer to this question. So we will discuss the other 3 as well to understand what is happening in the other 3. So in a freeze drying process, particles with maximum retention of volatile compounds is found in comparison to spray drying. So what happens in a freeze drying process is you have an oil dissolved in water or some kind of an emulsion, and that emulsion is frozen, and that frozen material is then passed on to the into the dryer, where then you will get a properly micro encapsulated product. So when you compare freeze drying and spray drying, if you have a lot of volatile compounds in your product, then best is to go for freeze drying, because that happens to retain most of the volatile compounds when compared to that of the spray drying process. So third is the pan coating, and pan coating is actually one of the most oldest industrial processes to form small particles or tablets. So what happens is you have a drum-like equipment, and or basically a pan rather. So inside the pan you have all your core particles present, and that pan will have a warm flow of air as well. So inside a drum or a pan-like structure, you have your core particles that need to be encapsulated, and then inside the pan you have a sprayer or an atomizer, where that will spray these wall materials on top of the core particles. So basically you will have warm air and the particles inside the pan, and then you will have your wall materials being sprayed onto the particles. And because of the warm air, what happens is that whatever wall materials is sprayed onto the particles dries and forms a coated particle. So that is what happens in a pan coating. So a more detailed explanation with all the figures and everything is given in the lecture 37. So if you guys want to have a more detailed explanation for this, you can go visit that lecture as well. And last one is the vibrational nozzle. So what happens is that you have an emulsion, a fluid stream of both liquid core as well as the wall material, and that is pumped through the concentric tubes, and what happens is there's a continuous vibration that happens, and because of this continuous vibration you have small droplets forming of the fluid stream, and this fluid stream will have already both your core material as well as your shell material. So basically if you want to divide them into drops, there is a continuous vibration that happens, and because of this vibration you have droplet formation and hence microencapsulation. So the answer is option A. Spray dryer is actually one of the most commonly used microencapsulation process in the food industry. Moving to question 7. Identify the correct option depicting the components or sections for a fluidized wet dryer in a Worcester process for microencapsulation. So this entire process of a fluidized wet dryer, also known as an air suspension process, is based on something known as a Worcester process. So Worcester is the scientist who kind of developed this process. So the fluidized bed dryer or the air suspension is also known as the Worcester process of encapsulation. So here you can see this is the equipment for the fluidized bed dryer, and you have 4 different sections. So we need to identify what these 4 sections are. So in this case, I'll briefly explain what is happening here, and based on that we can demark the different zones. So here you have process air that is coming in, and this air kind of moves in an upward direction. So you have process air coming in from the bottom and that moves to the top. And then you have the starting material that contains both your core material as well as your wall material. So your material will come in this direction. So the material will come in this direction and your air is moving in the upward direction. So because of this movement of air in the upward direction, your particles that are now being coated in this region will start to move upwards because of the air velocity. And at a certain stage, what will happen is that the air velocity, because of this expansion, will decrease. Now, if the air velocity suddenly decreases, what will happen is it will not have enough force to carry all the particles upwards. So certain particles that are here right now will start to fall down as well. This is because your air velocity reduces in this particular zone. Because again I'm repeating, because your air velocity decreases, the particles that was now carried with the flow of the air will start to drop down. So because of this, you are having kind of a segregation process that is happening here. So certain particles will move up, certain particles will come down, and this segregation process is necessary because if too many particles are coming together, then they might often stick together, they might agglomerate and then form a bigger size. They will not form a micro encapsulation, they will form large molecules. So to prevent this sticking together or the formation of large molecules or agglomeration, we need to start separating the particles slowly. So this separation is often enhanced by the reduced velocity of air. So if air velocity reduces at this stage, some of the particles will start coming down and some particles will still remain in the air. So because of this, now you're kind of starting to segregate the particles, and we're starting to segregate the particles that will prevent agglomeration. So one other important function for this zone is that because of air velocity moving here, your wall material that was coated onto the core particle will start drying because of the air that you are passing through here. So the air performs both the functions. It will first segregate also, and it will dry up your wall material. So all the solvent that was evaporated. So you have your wall material that has your polymer dissolved in a solvent. So what you want is that the product should be surrounded by the polymer alone, not with the solvent. So you need to evaporate the solvent. So this evaporation of the solvent will happen because you're passing air through this region. So because you're passing air through this region, the solvent will evaporate, and whatever particle was now coated will start coming down, and this is where you'll collect your final product. So again I'm just summarizing what is happening. You pass in air through the bottom, you pass in your other raw materials, that is both your core material and your wall material. Because of the air, your particles will start rising, and once it starts rising, suddenly there's a decrease in your air velocity. This will both because of the air, this will both dry up your wall material, and it will also start the segregation process where certain particles will start coming down. So it's both segregation happening as well as drying process happening after your air velocity has decreased. And at certain point, at this stage, what will happen is your solvent that was present in the particles will start evaporating, and what you'll get is your pure product with only the polymer coat around. So if you look at it, then this is where the fourth region is where you're sending in your air. So that is your inlet air part. So fourth is your inlet air part. Third is where you are sending in your starting material, that is both your core material as well as your wall material. So this is where majority of this is where both the particles will come in contact, both with the air as well as with each other. So this is where your main process is happening of your wall material being coated on top of your core material. So the third part is known as the process chamber. And the second part is where you see, because of the expansion in the valve, there will be a drop in the air velocity. So this part is known as the expansion zone. This is where your air velocity will decrease because of the expansion. So second is your expansion zone, and because of the expansion zone, certain particles will start coming down and will also be dried in the process, and you'll get your final product here, whereas your solvent will evaporate to the upper region. So this is where your segregation zone will happen. You're segregating both the molecules as well as the solvent from the molecule. So first is your, we'll start from the bottom process. First is your air inlet. Second is where the process happens, which is your process chamber. Sorry, third is where your process chamber happens. Second is your expansion region. And first is your segregation zone. So the answer to this question is option A. Okay, question A. Which of the following is the physical method for microencapsulation? So A, conservation, B, solvent evaporation, C, inotropic gelation, and D, air suspension. So there are multiple ways in which you can perform microencapsulation. There are three major classifications for it. First is the physical method, second is the physico-chemical method, and third is the chemical method. So under these classifications, you have a lot more other methods coming in. So which among the following is a physical method for microencapsulation? So you have A, conservation, B, solvent evaporation, C, inotropic gelation, and D, air suspension. So as I mentioned in the previous slide, what we discussed in the previous slide is also known as an air suspension. So it is also known as fluidized bed dryer as well as an air suspension. And if you can see here, none of these processes is dependent upon the chemical properties of your core or your warm material. This is basically just a physical process where you're supplying air, you're mixing both, and then you're making the particles drop after evaporation of the solvent. So this entire process is based on the physical nature of the particles and not based on the chemical nature. So air suspension is the answer for this. So air suspension is the physical method, whereas conservation and inotropic gelation are both physico-chemical techniques, where both of them depend upon your charges on your particles. So in case of inotropic gelation, what happens is your cation or anions that are present in the substances interact with the ionic polymers to generate a highly cross-linked structure. So the cation or anion will interact with the polymers to form a microencapsulation. That is what happens in inotropic gelation. Whereas in case of conservation, you have an electrostatically driven liquid-liquid phase separation. So liquid-liquid phase separation is suddenly you'll have droplet-like formation, and this droplet-like formation is because of electrostatics. That is, you're positively charged, any kind of a molecule. It need not be just your protein molecules or so. Even in case of other macromolecules, your positively charged molecule will interact with a negatively charged molecule, and because of this oppositely charged interaction, you will have droplet-like formation and liquid-liquid phase separation. So this is what happens in a conservation. So both these, if you see, inotropic gelation and conservation, this depend upon both the physical properties as well as the chemical properties of your product. So conservation and inotropic gelation are the physico-chemical techniques, whereas solvent evaporation is a chemical technique where you'll have a formation of a solution or a dispersion with your core material in an aqueous solvent. So core material that needs to be encapsulated is present in an aqueous solvent, and then you add some sort of stabilizer or so to form an emulsion, and once this emulsion is formed, you start to remove the solvent by evaporation. So this is what happens in a solvent evaporation process. The solvent evaporation process is a strictly chemical process, whereas air suspension that we discussed in detail in the previous slide is a physical technique for microencapsulation. The answer is option D. Question 9. Match the coating type with the most appropriate example used for microencapsulation. So you have in column 1 water soluble resin, second water insoluble resin, third wax and lipid, and fourth are proteins. And in the other column you have silicon, pea protein, methyl cellulose as well as stearic acid. So each one of these are examples for the different kinds of coating that can be given to for microencapsulation. So a water soluble resin here is the methyl cellulose. So methyl cellulose is a water soluble resin and this is water soluble at low temperatures. Whereas at high temperatures it starts to form a gel like structure. So based on the temperature you can actually say whether methyl cellulose is water soluble or not. But generally methyl cellulose is considered as water soluble at low temperatures. Whereas water insoluble resin is the silicon resin. So if you can see here, silicon is water insoluble. In fact, it can absorb water. So if you remember, silicons are you could even see in some kind of a packaging material where you have silicon packets given because they have the ability to absorb water that is present in the environment. So silicon is basically a very porous structure that has the ability to absorb water and it is not soluble in water. So silicon is in water insoluble resin. So the so third one, wax and lipid. So wax and lipids example is stearic acid. So stearic acid is a long chain fatty acid which comes under lipid category. So stearic acid is an example of wax and lipid coating type for microencapsulation. And pea protein, as the name suggests, is an example of protein. So methyl cellulose corresponds to water soluble resin. Silicon corresponds to water insoluble resin. Stearic acid is an example of wax and lipid. And pea protein is an example of proteins. So the answer here is 1 to 3, 2 to 1, and then 3 to 4 as well as 4 to 2. So the answer here is option C. Okay, question 10. Match the sentences given with the respective processes in autooxidation of lipids. First, the accumulation of peroxyl radicals to form non-radical products. Second, hydrogen reacts with molecular oxygen to form peroxyl radicals. Third, unsaturated lipids lose a hydrogen radical to form lipid-free radicals. So these three are the description of the steps, and the steps here are initiation, propagation, and termination. So in any kind of an oxidation process that involves free radical formation, these three are the processes that take place. First is the initiation or formation of a radical. Second is the propagation, where you have chain reaction happening. One radical will react with something else to form some more radicals. So you'll have a chain reaction happening in propagation step. And the termination process, often two radicals can come together to form a non-radical product. So this is how the entire reaction will be terminated when you have no more free radicals present. So we have to match this with the description of the processes. So first process is the initiation process. So in the presence of certain initiators, what happens is there are unsaturated lipids, or basically unsaturated lipids are the lipids that have double bonds in them, and those kind of tend to undergo auto-oxidation processes. So unsaturated lipids which have a double bond lose a hydrogen radical to form a lipid-free radical. So RH, which does not have any unpaired electron, in the presence of initiators undergo free radical formation, where you have R dot plus H dot. So both R dot and H dot will have an unpaired electron, which is why they are very, very reactive and they are very less stable also. So they immediately react with something else. So that is how your propagation step starts in. First is the formation of the free radical, and since these free radicals are extremely unstable, they start reacting with something else to start propagating the reaction. So the second step is propagation. So if you can link this, initiation corresponds to the third point, where unsaturated lipids lose a hydrogen radical to form lipid-free radicals. So this is your initiation process. So after this is your propagation step. So in the propagation step, your alkyl radical of unsaturated lipids, that is your R dot that was formed in the initiation step, contains a labile hydrogen, or that is a very unstable hydrogen that reacts very rapidly with molecular oxygen to form peroxyl radicals. So that is this reaction. So R dot plus O2 gives ROO dot. Here dot means an unpaired electron. So this step is much faster than its reaction with further unsaturated lipids to form hydroperoxides. So these radicals that are formed in the initiation step can either react with molecular oxygen or it can react with more unsaturated lipids. So this step is found to be more faster than this step. So you can either have it reacting with oxygen, and this will further react with other unsaturated lipids, and hence you can get a chain-like reaction forming. So again you'll have an R dot. So this R dot again will react with oxygen. So this is kind of a chain reaction that is happening that will lead to a propagation of your free radicals present in your oil and other compounds. So as you can see here, the hydrogen reaction with the molecular oxygen to form peroxyl radicals is the propagation step, because your peroxyl radicals will start interacting with further unsaturated lipids and cause this chain reaction. So your second corresponds to propagation, and which means your termination corresponds to accumulation of peroxyl radicals to form non-radical products. So at a certain stage, when your propagation is happening, at the last stages of the auto-oxidations, your peroxyl radicals will start to accumulate. That is, you'll have so much of radicals present that they'll start interacting with each other to form a product that is no more having an unpaired electron. That is, they are non-radicals and are not very reactive anymore. So when your peroxyl radicals start to interact with each other, they form non-radical products, and hence there is no more reaction to take place. There is no more free radical present in the food product, and hence your entire oxidation process terminates. So that corresponds to accumulation of peroxyl radicals to form non-radical products. So if you can see here, first step is your unsaturated lipids losing a hydrogen radical to form lipid-free radicals. These lipid-free radicals will have a labile hydrogen that reacts with molecular oxygen to form peroxyl radicals that corresponds to propagation. And then when you have too much of peroxyl radicals present in your food compound, these radicals will start interacting with each other to form non-radical products. So that comes under termination. So 3 corresponds to 1 and 2 corresponds to 2, and then 1 corresponds to 3. So your answer to this question is option C. Okay, moving to question 11. The effective interaction between the tocopherol and the citric acid in soybean oil is termed as A. rancidification, B. auto-oxidation, C. synergism, and D. chelation. So as I had mentioned before, tocopherol is an antioxidant. So it does not involve in auto-oxidation or rancidification. It in fact does the opposite. It acts like an antioxidant. What happens is when you're having oils and lipids, majority of them are unsaturated fatty acids. That is, they have double bond. So these double bonds undergo at certain conditions in the presence of oxygen undergo oxidation. So this is known as an auto-oxidation processes. So in the auto-oxidation process, what happens is your fatty acids start to liberate volatile compounds such as aldehydes and ketones. So the presence of these aldehydes and ketones give it a very off flavor or an undesired odor or color, and this is actually known as rancidification of oil. So basically your double bonds present in your unsaturated fatty acids will undergo auto-oxidation, and this auto-oxidation produces compounds like aldehydes and ketones, and these aldehydes and ketones result in an off flavor and off odor that leads to rancidification. So these two are undesired processes in an oil product. So you often add antioxidants to the oil to prevent these processes. So what happens is that often you can add multiple antioxidants together, and these multiple antioxidants work in a synergistic manner. So they have much more power or much more capability to prevent oxidation in combination rather than using it as a single product. So basically, instead of adding just tocopherol or just citric acid, which has lesser efficiency to prevent oxidation, you can add these in a combined manner to have a better uh, better prevention action, so or better activity. So because you're adding now in combination, the amount of tocopherol and the amount of citric acid that you need to add is also less. So with lesser amount of food products, you can get a much better activity against oxidation. So that is the entire concept of synergism. So antioxidant combinations are known to impart more protection against lipid oxidation than the sum of the individual activities of the components when used separately. So instead of using separately, if you add it in combinations, that imparts more protection. So this effective synergistic inhibition of autooxidation is known as synergism. And so vegetable oils containing tocopherol mixtures are effectively stabilized by adding citric acid. So basically, if you add tocopherol and citric acid, that has a better ability to prevent lipid oxidation when compared to just tocopherol or just citric acid. So this is an example of synergism. So chelation is also a way in which you can prevent oxidation, but that is majorly involved by adding some kind of a chelator molecule, such as just citric acid or EDTA. Those binds to metal ions or minerals present in the lipid molecules and prevent oxidation. So tocopherol is not a chelator. Only citric acid comes under chelation. So when you add both tocopherol and citric acid, which has two different mechanisms, you have a synergistic effect that happens. So the answer is option C. Moving to question 12. Which of the following coating materials is thermo-irreversible in nature? So you have A. Gelatin, B. Alginate, C. Agar, and D. Pectin. So one of the common properties amongst all these four are that their ability to form gel-like structures or kind of yeah, they all four of these have an ability to form gel-like structures. But this happens only when you start heating it. So you have to heat all these products to a higher temperature, and then when you cool it down, your cross polymerization starts to happen, and then you have a gel-like structure. So you have to heat it first and then cool it down. So often what happens is that when you cool it down, you get a gel-like structure. It can sometimes be reversible. That is, if you heat the gel again, it will again form a solution-like structure, a solution-like material. So sometimes this can be changed back into a solution form if you heat it. Sometimes it cannot be. So this nature, when you cannot reverse back the solution by heating it, it is known as a thermo-irreversible process. That is, first you heat it and then cool it down to form a gel-like structure. This gel, if you again heat it, has the ability to form a solution-like structure, but only in certain products. So we'll see which of these products are there. Gelatin, agar and pectin are soluble in water when heated, and this solution made from these wall materials are set on cooling. That is, it forms a gel-like structure upon cooling the mixture to room temperature. But this can be reversed back if you heat it again. Whereas in case of an alginate, which is a polysaccharide derived from seaweed, these often form complex with calcium. So when you're dealing with a coating material, generally we add calcium alginate, not just alginate. So the calcium alginate kinds of forms a complex, and this complex cannot be reversed upon heating. So calcium alginate is thermo-irreversible coating material, whereas gelatin, agar and pectin can be formed into a solution-like structure, solution-like uh, solution-like property upon heating it. So the answer is option B. That is, upon heating, gelatin, agar and pectin can be reversed back into solution state, whereas alginate cannot because of its complex with calcium. Moving to question 13. Dash is a type of step growth polymerization where two reactants react rapidly due to polycondensation. So A, you have cross-link polymerization, B, in situ polymerization, C, interfacial polymerization, and D, matrix polymerization. So these are different kinds of polymerization steps that happen. Uh, these are chemical methods of microencapsulation. So all these come under the chemical processes of microencapsulation. And all these differ in the method in which we generate your encapsulated products. So when direct polymerization of a single monomer is carried out on the particle surface, it is known as in situ polymerization. So here if you see this diagram, you have your polymer material dissolved in your continuous phase. So this is your continuous phase and where you're adding your oil or the product that needs to be encapsulated. So here you have in your continuous phase you have your polymer material, and the moment you add oil you form an emulsion, and in this emulsion what happens is your coat or the wall coat starts to accumulate around or polymerize around your oil. So then you have an encapsulated product. But these both of these seem very similar. That is, your in situ polymerization and your interfacial polymerization seem very similar. But one main difference is that here the coat material is present in the continuous phase, whereas here it is present both. One material is present here in the continuous phase, the other material is present along with the oil. So when you add in the interfacial polymerization, you have two different reactants in different phases. One in the continuous phase, the other one in the oil. So when you mix these two, then you form an emulsion, and whatever cross polymerization happens does not happen in the continuous phase. It happens at the interface of the two phases. That is, at the interface, at the border of the oil and the water is where the polymerization will happen. So this is known as the interfacial polymerization. Whereas in case of in situ polymerization, this will happen in the continuous phase. Whereas in this case, it will happen at the interface. So that is the difference between both the kinds of methods. So in the interfacial polymerization is a kind of a step growth polymerization where two reactants, both in this and this, will start to react rapidly due to polycondensation. So that is an interfacial polymerization example. Whereas in case of matrix polymerization, the core material, which is often porous, the core material is embedded in a polymeric matrix, and that matrix is porous in nature. So you have often beads, or let's say even calcium alginate beads are very porous in nature, and this polymeric matrix, the porous nature of the polymeric matrix can be exploited to embed your core materials inside it. So it will be like a spherical like structure with multiple pores inside it, and within the pores, inside the pores, you'll have the core material embedded. So that is known as a matrix polymerization. So here in this question, the answer is that the interfacial polymerization is the process where two different reactants react rapidly at the interface due to polycondensation. The answer is option C. So question 14. So this is numerical. 100 ml oil in water emulsion is prepared by mixing 30 ml of oil in 70 ml of water starch solution. So the density of the oil is given as 0.91 gram per centimeter cube. The emulsion is further converted into microencapsulated oil powder by spray drying with 110 degrees Celsius inlet temperature. Calculate the microencapsulation efficiency of the oil if the surface oil for 3 grams of oil powder is 0.5 grams. So in this case, when you're doing a spray drying process, that is with the help of an atomizer, you are spraying your oil water emulsion. What happens is that because of the high temperature, often you will have this microencapsulated product, but then your efficiency will not be 100%. That is, still some oil will be present on the surface, it will not be completely encapsulated. So at that stage, if your oil is still present on the surface, that means your microencapsulation efficiency has decreased. That is, all the oil should be present inside the encapsulation, not on the surface. The moment you have oil on the surface, it means that your microencapsulation efficiency is not 100%. So here it is given that for if you take 3 grams of your sample, that is your encapsulated sample, 0.5 grams of oil is present on the surface. So 3 grams of oil powder, in 3 grams of oil powder, 0.5 gram of oil is present on the surface. That is, it is not encapsulated completely. So now you have to find out what is the encapsulation efficiency in this case. So we'll first discuss the given here. Given the total volume of the emulsion is 100 ml, and in that 100 ml, volume of the oil is 30 ml. Density of the oil is given as 0.91 gram per centimeter cube or gram per ml. The amount of surface oil present after microencapsulation is 0.5 gram per 3 gram of oil powder sample. So this is the given. So first we need to calculate, it is given for every 3 grams. So for the entire product, the entire emulsion that we had microencapsulated, how much amount of surface oil is present is what we need to calculate. So based on that, we will calculate how efficient the process was. So total mass of oil initially taken for the microencapsulation process is density into volume. So basically mass by volume is density. So you already know what is the volume of oil that you took initially and you know the density. So from this you can calculate what is the total mass of oil you took. So the total mass of the oil that we had taken for the microencapsulation process can be given as density into volume. So that is 0.91 gram per ml into 30 ml. So this will give me the total amount or mass of the oil that was present in my water emulsion, oil water is emulsion, which is 27.3 grams. And then it is already given in the question that for every 3 gram sample, 0.5 gram of oil is present at the surface. So if in every 3 grams, 0.5 gram is present in the surface, then for 27.3 grams of oil that we took initially, how much amount of oil is present in the surface? So it's basically a unitary method. So if for 3 grams, 0.5 grams of oil is present at the surface, then for 27.3 grams of oil, the amount of surface oil present is 27.3 into 0.5 by 3 grams. That will give me 4.55 grams of surface oil. So this is basically unitary method. For every 3 grams, 0.5 grams present at the surface. So for 27.3 grams, you have 4.55 grams present at the surface. So 4.55 grams of oil was not encapsulated properly. So in order to find out the microencapsulation efficiency, that is equal to amount of oil encapsulated by initial oil amount into 100. So we know how much of oil was not encapsulated. So you can find out the oil encapsulated by subtracting initial oil minus oil that was not encapsulated. So again to repeat, surface oil is the oil that was not encapsulated. Initial oil is the total amount of oil that you took before starting the process. So initial oil minus the surface oil will give me amount of oil that was successfully encapsulated. So we know what is the initial oil, which is 27.3 grams that we calculated. We calculated what is the surface oil. From there we can calculate the efficiency. So 27.3 grams is the initial amount of oil that we took. 4.55 grams is the total amount of surface oil that is still present, that has not been encapsulated still. So that divided by initial amount again gives me an efficiency of 83.33%. So the answer to this question is option B. So this is how you calculate the efficiency of microencapsulation process. Moving to question 15. Match the given sentences with the respective processes. So first, the production of triangular prismatic shaped nanoparticles. Second, produced by interfacial polymerization and electron spinning. Third, produced by layer deposition technique. So these are the different production methods, and the second column has the final product. First is the nano biosensor, second is the nano laminates, and third is the nanofibers. So nano biosensors are the ones that are triangular prismatic shaped nanoparticles that can detect biological threats. So nano biosensors can detect antibodies, it can detect pathogens and so on. So nano biosensors can detect biological threats or biological samples or antibodies, proteins, pathogens, etc. So these nano biosensors are triangular prismatic shaped nanoparticles. And second, the nanofibers are produced by interfacial polymerization and electron spinning. So we discussed what an interfacial polymerization is. That is two different reactants, one in the continuous phase and other one in the other phase can be are mixed together, and the polymerization happens at the interface. So that is known as interfacial polymerization. And whereas electron spinning is a method where you charge your emulsion, that is you give your emulsion a certain charge And because of electric force, you can now drag these charged emulsion into fine thread-like structures as given here. So if you Google your electrospray mechanism, you'll have a charged solution coming out, and this charged solution will be dragged to form these thread-like structures according to the diameter that you want. So this is known as electron spinning, because you're charging your particle and using electric force, you're creating it into different dimensions. So this is known as electron spinning. So nanofibers are formed by interfacial polymerization and electron spinning, and this electrospin polymer fibers have unique mechanical, electrical as well as thermal properties. And these polymeric strands are of sub-micrometer diameters. So nanofibers correspond to interfacial polymerization and electron spinning. So next that we have are nanolaminates. So nanolaminates are basically two different kinds of compounds that are stacked one on top of the other. So basically they are layer by layer techniques. So first layer you have surfactants along with your active compounds. Then you have charged biopolymers. Then you have again surfactants. Then you have again polymers. So these are layer-like substances. They are formed layer by layer deposition, and this results in nanolaminates. So nanolaminates are basically two or more layers of materials with nanometer dimension, and these are either physically or chemically bonded to each other. So hence you are employing a layer by layer deposition technique. So this is how nanolaminates look like. So the first corresponds to biosensor. Electron spinning corresponds to nanofibers, that is 2 corresponds to 3, and 3 corresponds to 1. The answer is, 3 corresponds to 2, sorry. So your answer is option A. So question 16. Which of the following are the essential prerequisites for the formation of ketonic rancidity? A. Presence of molds. B. Presence of short chain fatty acids in the medium. C. A low partial pressure of oxygen. And D. All of these. So ketonic rancidity, there are different kinds of rancidity involved. You have oxidative rancidity or hydrolytic rancidity. A third one is the ketonic rancidity. So ketonic rancidity are formed by molds or basically bacteria and fungi. So these two are the ones responsible for the ketonic rancidity, because these molds and fungi have enzymes in them that result in oxidation process of the lipids. So what happens is that your fungi like Aspergillus niger, the enzymes in this fungi will start attacking your short chain fatty acids present in the lipids, and this, in the presence of low partial pressure of oxygen, will result in formation of ketones and other products that give it a certain off color and an off taste. So because of this ketonic products, you will have your oil turning into an undesirable property, that is possessing undesirable properties. That is, you will have an off color, very dull color, and have a very off flavor and taste as well. So this happens because the enzymes present in your molds or your fungi starts attacking the short chain fatty acids at a low water activity and low partial pressure oxygen condition. So all of these are essential prerequisites for the formation of ketone rancidity. The answer is option D. So this ketonic rancidity results in undesired properties of your oils. Okay, moving to question 17. Match the antioxidant types with their suitable example or function. So here you have different kinds of antioxidants. First, synthetic antioxidants. Second, secondary antioxidants. Third, metal chelators. Fourth, oxygen scavengers. And fifth, natural antioxidants. So these are the different kinds of antioxidant types. And in the second column, you have the example or the function of those corresponding antioxidants. So you have beta carotene, propyl gallate, regeneration of tocopherols, fourth, prevention of metal redox cycling, and fifth is ascorbic acid. So we'll start with the first one. So an example of the synthetic antioxidant is propyl gallate. So amongst all these four, as you can see, amongst beta carotene, propyl gallate and ascorbic acid, propyl gallate is the synthetic antioxidant. Then you have secondary antioxidants coming in. The secondary antioxidants, one of the functions of the secondary antioxidants is the regeneration of tocopherols. So your tocopherols are primary antioxidants, and in order to regenerate these tocopherols, once their antioxidant property is done, we add secondary antioxidants. So these secondary antioxidants will regenerate the primary antioxidants so that it can still perform its function. So secondary antioxidants have the function, one of the function of regenerating tocopherols. Third, metal chelators, which are examples of secondary antioxidants, perform its function by prevention of metal redox cycling. So metal chelators have the ability to form, to sequester metal ions, and because you are sequestering metal ions, this prevents the metal redox cycling. So 3 corresponds to 4, and then you have oxygen scavengers. So oxygen scavengers, actually both beta carotene and ascorbic acid are natural antioxidants, but only ascorbic acid functions by the method of oxygen scavengers, whereas beta carotene has a different approach. So ascorbic acid is an oxygen scavenger, whereas both natural antioxidants, that is the natural antioxidants, are both beta carotene as well as ascorbic acid. So here you have synthetic antioxidants. 1 corresponds to 2, and then 2 corresponds to 3, and then 3 corresponds to 4, 4 corresponds to 5, and then 5 corresponds to 1. So the answer is option A. Question 18. Which of the following statement is true for antioxidants? The safety of natural antioxidants are known in most cases. B. The surfactants have no effect on partitioning properties of the antioxidants. C. Polar antioxidants are more active in bulk oil systems. And D. The ideal antioxidants are readily absorbed by the body. We'll start with the first statement. The safety of the natural antioxidants are in fact not known in most of the cases. So we don't know what kind of effect or long-term effect these natural antioxidants can cause. So we don't know the safety of these natural antioxidants, but generally if they come under the GRAS category, that is generally recognized as safe. If they come under the GRAS category, then we need not do any safety test. We can assume that the antioxidants are safe. But in reality, the safety of the natural antioxidants are not known for most of the cases. Second, the surfactants do have an effect on the partitioning properties of antioxidants. So you're adding an antioxidant to an emulsion process, an emulsion or so. So it needs to partition between your oil and your water and so on. So this partitioning properties of antioxidants, certain antioxidants function at the interface of oil and water. So all these functioning ability of the antioxidants depends upon how well it is able to partition between oil and water. So the partition properties of antioxidants are dependent on multiple factors. So the factors include the chemical structure and the polarity of the antioxidant, the type of the lipid substrate that you're dealing with, the kind of oil that you're going to put it inside, the presence of surfactants, as well as the composition of the bases. So all these factors determine the partitioning properties of the antioxidants. So B is also false. Surfactants do have an effect on the partitioning properties of antioxidants. And C, polar antioxidants are more active in bulk oil systems, which is true. So this is known as a polar antioxidation paradox, where it was generally found after research studies that polar antioxidants in general were found to be more active in bulk oil systems. So C is true, whereas D is false. So you, in an ideal antioxidant system, you do not want it to be readily absorbed by the body. So ideal antioxidants are not absorbable by the body. So out of all these 4, only the third option is correct. So answer is option C. Question 19. Which of the following shows the correct order of polarity among given synthetic free radical scavengers? So you have propyl gallate, you have BHA, you have BHT, and you have TBHQ. So based on the chemical structures or the chemical formula, the polarity of these four free radical scavengers or antioxidants are different. So amongst these, all of these are synthetic free radical scavengers. So among these free radical scavengers, the most non-polar is the butylated hydroxytoluene, which is BHT. Butylated hydroxytoluene is the most non-polar, followed by BHA, which is butylated hydroxyanisole. Butylated hydroxyanisole, and then comes the tertiary butyl hydroquinone, TBHQ. And then the least non-polar amongst these is the propyl gallate. So the D is the option. First is butylated hydroxytoluene, followed by butylated hydroxyanisole, then tertiary butyl hydroquinone, and then propyl gallate. The answer is option D. Question 20. Which of the following nanolithography techniques employ a tightly focused beam of electrons scanned over the surface of a substrate? So you have photolithography, X-ray lithography, E-beam lithography, and soft lithography. So before we proceed with this, we'll discuss what a lithography is. So the lithography is basically a process where you create patterns of desired designs. That is, you'll have a certain design based on what kind of application that you have for that substrate. So these patterns of designs are first transferred onto a base substrate, mostly through masks. So these masks will have the will be on top of your substrate, and you will pass multiple and you will have different methods of creating these patterns. So these are the different methods in which you can create patterns. You can either have a photon based method, you can have an X-ray based method, you can have an electron beam based method, and you also have something known as a soft lithography. But in all these processes, what happens is you create or you design certain patterns onto your substrate, and this is known as lithography. So a photolithography consists of a patterning substrate by employing the interaction of beams of photons or particles with material. So basically photolithography involves light or photons that is passed to create these designs onto your materials. This is used in integrated circuits manufacturing, photolithography. And as the name suggests, X-ray lithography uses X-rays to create geometric patterns uh, to a light sensitive material. So in case if you cannot use a light photolithography, that is in case of light sensitive material, you can often use X-rays to create these designs, and this is often used in transistors. So in E-beam lithography is where you use a tight beam of electrons, and these electrons will be scanned over the surface of the substrate, and this helps in creation of the design that is required. So electron beam lithography uses a tightly focused beam of electrons scanned over the surface of a substrate. Typically, the electron beam lithography with ultra high resolution is used at the very beginning of a multiple technique or multi-step process in a top-down approach in order to transfer the nanostructure onto the substrate and subsequently build up a device in a layer by layer fashion. So electron beam lithography uses the beam of electrons and is generally used as one of the first steps in nanostructure creation. And then you have soft lithography. So in soft lithography, you generally do not use these beams or so. So what happens is you use a very as the name suggests, soft lithography. You have a very soft material or a very elastic material. So thus elastomeric material is known as the PDMS or polydimethyl siloxane. So this is a very flexible and it's a very elastic material. So this elastic material will have patterns present on it. That is the pattern that you want. So on top of this PDMS, you will add your pre-polymer and then cure it. That is, you will set the polymer and then you will peel away the PDMS. So now your polymer will have this embossed layer of designs that was originally present in the PDMS. So here you are not passing any ray or laser or anything of that sort. It is a very soft technique where using an elastomeric material which already has a design, then you're pouring the polymer on top of it, then you're setting it and then removing away this lower surface. Then you have an embossed layer of polymer. So this is your soft lithography. The answer is option C. So when you require a nanolithography technique that requires a tightly focused beam of electrons, you use E-beam lithography. The answer is option C. So hope the problems were clear. If you guys have any doubts, you can email or you can post it in the discussion forum. We can repeat the questions again in the next class if you guys want. Thank you. So with this, we conclude Saturday's problem solving session, and we'll have the next session corresponding to week 9 syllabus the coming Saturday. Thank you.